So I think that's the trickle of people has slowed down, so I think we can get started. So yeah, good afternoon, everyone. Welcome to the last EPI seminar series of the semester, brought to you by the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health. So I'm James, I'm going to be moderating the session, and I'm really excited and honored to welcome with us today Professor Brian Bryson, who's an assistant professor of biological engineering at MIT. So I'll just go through a few logistics before we get started. So as you heard, we're recording the seminar today. So just really double check that your camera and microphone are off if you don't want to be recorded. And just to say, the recording will be available on YouTube at a later time. So I'll just introduce our speaker. So Professor Bryson is an Esther and Harold Edgerton Career Development Assistant Professor of Biological Engineering at MIT and the Reagan Institute. He obtained his PhD at MIT in Biological Engineering and completed postdoc training at the Harvard Chan School of Public Health, where he applied his engineering and computational skills to understand TB pathogenesis. So his lab's research interests now sit at the intersection of infectious disease, innate immunity, and systems biology, where they develop novel tools to predict bacterial fates, interrogate host-bacterium interactions at the single-cell resolution, and reprogramming the innate immune system to improve bacterial control. And more recently, Professor Bryson and his group have turned their computational skills to adapt machine learning methods to predict viral evolution and immune escape, which I won't say more about lest I spoil Professor Bryson's talk. So Professor Bryson has about 40 minutes for the talk, and then we'll stop for questions and discussion. If you have questions that come up through the talk, please do send them to Flavia Campanovo in the chat, and let us know if you want to be unmuted and ask the question yourself at the question session. So I think Flavia will send a message in the general chat. There you go. So that's all I have to say. So without further ado, thank you very much for joining us today, Professor Bryson, and please take it away whenever you're ready.
Awesome. Well, thank you, James, for the introduction, and thank you all for having me. I'm really excited to share with you some of the work we've been doing in my research group. I started it in 2018. That seems like such a long time ago, but I'm really excited to both share with you what we've been up to and have a lot of discussion about how we can use some of these tools that we've been deploying for the problems of infectious disease. And, to me, I was trying to think about how to kind of stitch together the two halves of my talk. And, as a microbiologist, an aspirational microbiologist is what I call myself, I think about the phrase that small changes can have big impacts. And that's going to be a real theme here in today's talk, and how we think about how individual pathogens can wreak havoc, but also about how small changes at the nucleotide level or at the amino acid level can give rise to either improved antibody control or diminished antibody control in the setting of infection. And so broadly, what my research group has evolved to do in the last 3 years is really think about how we bridge molecular immunology, bioengineering, and quantitative modeling to study host pathogen interactions. And a key question for us as we think about translational impact is how can we intervene in and disrupt infectious pathogens? And so today's talk is really separated into two halves, where the first half we're going to talk about how we might think about going after bacterial pathogens using antibiotics or novel small molecules, and the second half of the talk will be focused on how we can think about viral evolution and antibody immune escape. And so, so I probably don't need to remind this audience that tuberculosis is a huge global health problem. It's one of the leading causes of infectious disease death. 2 billion people are estimated to be infected, so about a quarter of the global population, 4,000 deaths per day. And that's probably likely been exacerbating in the setting of COVID disease. 1 in 10 individuals with active TB disease, and not all hope is lost, because we do have an antibiotic regimen that is effective for drug-sensitive TB, which is a 6 to 9 drug regimen. And what would be transformative for the field is a protective vaccine, but we don't yet have one that is licensed. So those are kind of the hurdles that we still confront for TB. And one of the questions that we have been really interested in asking is, when we think about intervening in the setting of an infectious pathogen, when we think about antibiotics, antibiotics have targets. And one of the things that we think about a lot is how can we think about those targets, namely proteins, and how we can disrupt their function in order to advance new therapeutics. And a tool that we have become big fans of in my lab are a set of computational tools called language models. And in particular, this is built off of really seminal work from the Berger group at MIT, where they kind of pursued this simple analogy, is that in this field of natural language processing, we can learn from a corpus of text and identify patterns using distributional semantics to understand how these different words kind of come together to form a sentence. So any of you who've ever sent a text message these days or used Gmail, these are the natural language processing algorithms, or what really help predict what is that next word in the sentence. And so an analogy in the computational biology field has been that instead of thinking about these tokens or words in English text, what you can instead think about are amino acids that really form the language of a protein. And so you can imagine using something like this FASTA file that I show you here in the middle that has just a bunch of different amino acid sequences for a given protein. But really the workhorse technology is really these computational models that really allow you to learn the grammar, or in our case, in the second half of the talk, also learn the semantics. And so what these language models really do is they are trained on a corpus of amino acid sequences. And traditionally it's done using some neural network framework. I won't go into more detail beyond that. And the idea is that the idea behind these models is you can give it an amino acid sequence of interest. And then what it will do ultimately is predict what the most likely amino acid is at a given position that you don't offer to the model. And so this is what the grammaticality of these models are. And so the analogy here is when you're thinking about sending your text message, and here's the next word that's being predicted, these natural language models applied to the setting of a protein are really trying to do that same exact thing. And so the hypothesis for us has been, given the preponderance of data that are now available for amino acid sequences, how can we leverage these protein models to develop novel insights for pathogens or pathogen-derived proteins that may be of pharmacological interest? And so that was the big question that we wanted to set out to ask. And so the first task that I'm going to tell you about is trying to target this protein in Mycobacterium tuberculosis. This is a protein kinase called PKNB. And an important task for us, especially in the infectious disease field, given the limited kind of industrial interests or biotech or pharma interests in TB and TB drugs these days, a big question has been, how do we begin to identify novel targets, or basically new small molecules that target an essential protein? So what do you need to know about PKNB? PKNB is an essential protein required for Mycobacterium tuberculosis survival. And so the hypothesis is that if we could actually begin to identify small molecules that could target this protein kinase, maybe we could begin to identify a novel set of compounds that could orthogonally come in and target Mycobacterium MTB survival. Now, what's the challenge? The challenge is, okay, can we actually use machine learning to do this? In part because of two problems. One problem is we have limited amount of data for here are known small molecules that actually bind to PKNB. And so if we think about all of the molecules that are out there, one of the challenges becomes, I'll tell you the truth. I almost failed organic chemistry as an undergrad. And so thinking about actually synthesizing a chemical compound really terrifies me. But what was really exciting is to think about how we could utilize these language models paired with really great featurization and representation of small molecules to begin to learn and say, could we just skip the ring structures or like the chemical synthesis and identify molecules that already exist? And what we really wanted to do in this problem was we wanted to say, can we find the right small molecule? In particular, in the way that we created this problem is that by right small molecule in this setting, what we wanted to find is a small molecule that would bind PKNB with a high affinity. And where the hypothesis here is that if you have something with high affinity, you need less drug, potentially fewer side effects. But a real problem, again, is that these compound libraries are incredibly large. And TB is incredibly slow, and working at BSL-3 is incredibly challenging. And so if you can imagine what you might be able to pull off in a pharma in a pharma setting where you have these large molecule liquid handling robot exercises that you can really just kind of brute force through a whole host of different small molecule and target compound libraries, that's really hard to do in a BSL-3 setting experimentally. And so what we wanted to ask specifically is, could we actually begin to perform what a pharma industry would do in the laboratory setting, all in silico? And the idea here is that what we wanted to do is really use these models to say, could we actually predict binding affinity as a function of, given a protein target of interest and a small molecule of interest, could we actually predict binding affinity? And if we could predict binding affinity, would those small molecules actually work to control MTB growth? And so here's the dirty laundry about machine learning that I think that we don't oftentimes address, is that especially in the biological setting, if I'm Google and I'm just learning from people who are just, doing web searches, that data is, one, it's cheap to obtain, and two, there's a lot of it. Now, by contrast, if we think about the biological setting, we often have much less training data. In particular, the data set that we actually used for this analysis was about 72 compounds and 400 kinases. And that's the training data we had for affinity. And so what happens when you give that information to a traditional off-the-shelf machine learning model? So here we can say, here's some f of x that we're trying to predict. And let's just imagine that that f of x here is affinity of a small molecule for a particular protein of interest. Now, if you imagine this red dotted line is what we'll call our ground truth. And this solid light blue line is what our machine learning model prediction is. Now, if we think about regimes in our training data that are very similar, where we have this observed domain, for example, oftentimes those predictions are very much aligned with what our ground truth observation is. Now, what happens with traditional off-the-shelf machine learning models is that when you go into this unobserved domain, oftentimes you're subject to pathological or overconfident predictions. And as biologists, one of the things that I think we oftentimes appreciate is that error bars give us some measure of uncertainty. And so the question we wanted to ask is, could we improve upon our machine learning models by taking one step further, and instead also have these models return a measure of uncertainty associated with a given prediction? Where the idea is that now, if you actually are empowered by both the information about the affinity, but also the associated uncertainty with the measurement, then you could yourself balance how much are you want to prioritize exploring potential new regimes that have a lot of uncertainty, or exploiting areas or exploiting other small molecules that are similar that then would ultimately probably be more likely to have the affinity that you desire. And so that was the kind of the machine learning principle that we wanted to pursue, is that we really wanted to be able to model uncertainty in these approaches. And to give you some intuition, you can think about playing the game Battleship. So for those of you who haven't played the game in a while, Battleship is a game of like trying to identify where your hit is. In this case, it's the hit of your battleship, for example. But initially, when you think about playing this game, what you have is your opponent has like aligned their battleships on this grid in some position, and you're trying to guess where that battleship is, where that hit is. But initially, you have no information to make a really exquisitely informed guess. So initially, you make your first guess, and you might say C3, and your opponent says miss. All right, so now let's go on to round 2, and then you say, all right, I'm going to pick F6, and your opponent says hit. And so what this does is this actually improves your kind of understanding or your kind of uncertainty about particular positions in this grid or this space that you're searching now. Where if you know like how this game is played, you know that your ship is either 2, 3, 4, or 5 units long. And so that tells you that, okay, in this first round where you got your first hit, now you have very low uncertainty about where that ship is most likely to be positioned. But as you think about the fact that you could be a 2-ship unit long, or you could be 4 ships long, as you move away from that initial hit, your uncertainty will increase. all right, so let's make our next guess, and you guess G6, and your opponent says miss. And so what this does is this actually helps improve your uncertainty about other positions, and it really also allows you to eliminate these other positions going to the right here, for example. So we can iteratively do this. You get another miss, you update your uncertainty estimates, and then you finally get a hit at F5. And so now you've got this kind of idea that, okay, I'm either going to be in a 4 or 5 unit ship, or that'll be the end of the game. And now your opponent says, you sunk my destroyer. All right, so we just played a board game, but what does that help us understand about the modeling approach that we can utilize here to understand the principles of uncertainty? And so we can think about what we did in this first stage, which is we did this round of exploration, where we're really looking for any potential hit. And then once your opponent says that you made your hit, now you can imagine this kind of exploitation regime. And what we wanted to do is like take these principles that you might imagine utilizing in a game like Battleship and apply that to the idea of small molecule discovery. And so what we wanted to do is we, in fact, wanted to model uncertainty using what's called a Gaussian process. And so now here, instead of just returning some f of x or your protein affinity for your small molecule in this case, what you'd imagine is that you return some affinity prediction, but you also have some measure of uncertainty associated with each one of these new predictions, with the idea that now by prioritizing both the affinity and the uncertainty, that would help guide your decision-making in identifying some new small molecules. So that was the task, that we wanted to now not only predict affinity, but we wanted to predict an associated uncertainty. And so what did we actually do? So we actually focused on PKNB, an essential kinase for MTB survival. We had access to publicly available data. Most of the experiments in this paper, in this talk I'm telling you about, were actually done, modeled initially almost on all publicly available data. So we had 72 compounds in a 440 molecule, small molecule kinase affinity prediction. And what we wanted to do is we wanted to predict the affinities for about 11,000 commercially available compounds. And so what these models do is, again, we're using these language models that really just are some featurization of a bunch of different amino acid features represented in numbers, but we also have to do this for a chemical structure. So what I'm showing you here on the left is actually when we reduce kind of this chemical structure into this linear vector and just project it down into 2 dimensions, we can see like here, each blue dot is one of the 11,000 compounds in the Cayman Chemical Library. And then all of these orange X's are just observations that were actually in our original training data. And what I want you to take away from this is that we have some some compounds that are very similar to the compounds that were originally in the training data, but we also have compounds over here that are very much structurally different from the training data. And so the idea here is that what we want to be able to do is actually now explore all of these blue dots to identify what are the most likely compounds that are going to have a high affinity for PKNB. Okay, so what we did is we actually applied the Gaussian process to this data. And what we asked the model to do is go ahead and say, all right, here's our training data. What is the associated uncertainty in any of these predictions? And what I hope you can see is that your uncertainty rises as you go away from your original training data. And so this is important because, again, what I said earlier is that a lot of these machine learning approaches are subject to pathological predictions. And so now at least we can actually model the uncertainty associated with any given small molecule or prediction. And then we can actually ask the model to tell us when the model says, okay, I'm going to acquire a small molecule. It says, go ahead, Brian, and order this small molecule. What you can see is that it prioritizes acquiring small molecules that are more similar to the original training data. Now, by contrast, if we use, say, for example, a multi-layer perceptron approach, this is actually subject to high pathology, you can say, for example, because what you can see is that it's actually saying, go ahead and select these small molecules that are very, very different from any of the training data that you even gave it to. So again, it's saying, go ahead and pick this one, even though it has really no justification for doing so. All right. So now the question is does this actually result in any improvement in our predictions or acquisition of small molecules with desired properties? And so what we did is we actually just did something very simple. And so what we said is, okay, let's go ahead and have the model predict both uncertainty and the affinity. And we just use a simple ranking, an upper confidence bound to say, okay, we're going to basically pick small molecules from this 11,000 small molecule library based off of the affinity prediction and the amount of uncertainty. But we also said we can weight our uncertainty to kind of decide how risky are we feeling. So what I want you to take away from this left graph here is now we actually obtained all these small molecules. We performed affinity measurements for pKnB. And what you can see is without modeling uncertainty, what you can see is that actually, or without uncertainty, what you can see is that even our kind of off-the-shelf Gaussian process-based model actually can return small molecules that have very high affinity for pKnB. Now, by contrast, there are these molecules that have higher than a 10 micromolar affinity. Now, if we look at a multi-layer perceptron, all of the small molecules that the multi-layer perceptron recommends had experimentally defined very low affinity for pKnB. Now, when we actually say, let's go ahead and prioritize and weight, give a weight to the affinity prediction or the uncertainty prediction, what you can see here is that when we actually weight our uncertainty with a beta of 1 or even of 20, what you can see is that has an improvement in the affinity. So importantly, the takeaway here is that actually by incorporating uncertainty into our predictions, we can actually do a better job of identifying small molecules with a defined affinity for our target of interest. So we did this for pKnB, and just really quickly, I just want to tell you that it generalizes. So then we sought to apply this to a whole host of other small molecules, or no, kinases of interest. And again, the punchline here is the same. When you prioritize affinity, prioritize uncertainty, what you do is you always return small molecules that have, on average, better affinity for their target than a multi-layer perceptron. So this is really exciting because what we were able to do is just use really a state-of-the-art language model that was able to kind of represent pKnB in a set of vectors that really kind of captured the important features, and then paired with this featurization of the small molecule, we were able to identify small molecules of interest that that actually bind pKnB. And maybe because in the interest of time, I really want to talk about the other half of our work, I'll just kind of jump to some of the interesting punchlines. And so one question you might ask is, these pKnB drugs that you identified, did they have any bioactivity against intact MTB? The answer is yes. Actually, one of the small molecules that we identified is actually a lead compound that other people are now working on for pKnB targeting. So what was exciting is that my computer science graduate student was able to kind of recapitulate expert biochemistry, having never taken biochemistry himself. So to conclude maybe the first half of this talk, what I hope to have shown you is that incorporating uncertainty in machine learning algorithms for biology can really augment small molecule discovery, and that these small molecules that we identified have high affinity for the target protein of interest. And as I mentioned to you, they have whole cell activity. And also in this paper, what we showed is that this kind of principle of incorporating uncertainty is not limited to the problem of small molecule binding to a protein kinase of interest. We actually show that this kind of principle, that these principles can be extended to protein engineering and gene editing. Now, what made this all successful, again, were these really state-of-the-art language models that we can apply to understand how we might represent a particular protein using kind of a mathematical representation. And so for the second half of the talk, I really want to return to this idea of how do we intervene in and disrupt infectious pathogens. Now, again, we're going to be using the similar tools of language models, but again, what we're going to do here is we're going to apply these language models in a new way to understand viral escape and evolution. And so it's not lost on me that even though we're like literally maybe 3 miles away from each other, or like MIT is 3 miles from HSPH, I can't be with you in person because we're in the middle of this pandemic, which, you know, I think just underscores the importance of the work that we do, and the importance of really trying to apply all of the knowledge that we have available to understanding infectious disease. And so the question that we wanted to ask in this work was, could we actually build upon these protein language models to develop new insights? And in particular, what we really wanted to understand is this idea of semantic change or evolution. And what these algorithms do, these natural language algorithms do at their core, is what they learn is this concept called distributional semantics. And this is an idea that was actually proposed in linguistics in the 1950s, where the idea is that you shall know a word by the company it keeps. So if you are an algorithm, what you're doing is you're reading every news headline or every email that everybody sends, and really looking at the co-occurrence between individual words in a sentence and their order and how frequent that occurs to really build these distributional profiles. And what we reasoned is that maybe we could actually apply this to the problem of viral evolution. But at the core, before I go on to more details about that, just to give you a sense, again, for how these language models operate, what a language model does is it learns the probability of a word given its context. So we can take this phrase, the Chinese president blinked to Japan yesterday, and in the setting of English text, what the model might return is that the probability of the word went, filling in this blank, has a probability of 50%. But really importantly, when we think about evolution or change, the important other thing to realize is that small changes can have big semantic effects. And so we can start with the sentence, the boy pats the dog. And so if we change that original A to an E, the sentence becomes the boy pets the dog. Now, what's the takeaway here? This new sentence, the boy pets the dog, is grammatically correct and has semantically similar meaning to the original sentence. But again, just a single token change or a single letter change in this setting. Now we can change that original P to an E and the sentence becomes the boy eats the dog. This is still a grammatical sentence. It might not make sense, but it is very different in meaning from the original sentence. And so again, this was the conceptual idea when we thought about the natural language analog is like, okay, there are kind of conceptual parallels between how we might think about how a sentence might change as a consequence of changing individual letters, and how we might think about how proteins might evolve or how viruses may evolve in order to look different to the immune system. And really that was kind of the conceptual idea that we pursued in all of this work. And so what we proposed was this idea that we could augment natural language processing algorithms to pursue this idea of a constrained semantic change search. And so what you're given is a sequence of tokens from some language. And the goal is to find a single token change that, one, induces the largest amount of semantic changes learned by the model, and two, is constrained by the rules or grammar of that language. And so thus far, I've shown you examples of English, sequences of words from English, but the hypothesis is that we could apply this to understand sequences of amino acids from a viral protein. And this is when I tell you that, I come from a family of science non-practitioners and, but enthusiasts. And, when I talk to my grandma about viral evolution, because she's glued to the news, she's like, how do I think about this? And I was like, if I'm a virus, what do I want to do? I want to evolve in a way that allows me to look different to the immune system, but I still need to preserve some of my key functions. For example, like ACE2 binding or other receptor binding events. And so that was kind of the idea that we pursued, is that perhaps grammar might represent some of the key functions required for survival, and that semantic change might actually represent some element of looking different to the immune system. But before I go on and tell you the biological results, let's just give you some more intuition for how this algorithm works. And so for those of you who are machine learning aficionados, basically what we're using is a state-of-the-art neural network-based model. We're using a bidirectional long short-term memory neural network. And the important thing to know about that is that allows us to read both from left to right as well as right to left. Whereas by contrast, in English language, you might only be thinking about learning patterns from left to right. This bidirectional model lets us learn from each direction. And so what these you have this, what we consider this output of our model, which we hypothesize would represent grammaticality, as the field has considered for a very long time. But all these hidden layers would be some element of semantic representation. And so that was the hypothesis going in. And so now to give you a little bit of flavor for how constrained semantic change works, specifically on English text, we can go to this original headline. And actually, when the graduate student did all this work, what he actually did is he actually trained on a corpus of news headlines acquired from, like, let's say the 1950s to 2009 in Australia. And he said, okay, let's go ahead and apply constrained semantic change search to this phrase, wine growers revel in good season. Okay, so the first thing you wanted to ask is, let's go ahead and ask for a single token change. So in this case, a single word change to this headline that is semantically closest, but still constrained by the rules of grammar. And what the algorithm actually proposes is wine growers revel in strong season. So hopefully this feels to you semantically similar to the original. Now, by contrast, we can ask the algorithm to propose the most semantically different phrase possible it can think of. And what it actually proposes, and I just love this one, is wine growers revel in flu season. All right, so that was kind of the conceptual model when we think about English language. But importantly, when we thought about this from the perspective of a virus, we could imagine something very similar occurring, where the idea is that what we wanted to do is we wanted to model viral escape as semantic change, where you could think about influenza HA, for example, where this single histidine to serine mutation now renders this broadly neutralizing antibody C179 unable to bind HA. So again, small changes having big semantic effects, but now through the lens of protein structure and antibody binding. So in order to really pursue this hypothesis even further, what we wanted to first ask is, okay, what does a language model trained on sequences alone learn for semantics for a protein? So this is how this works. So what we did is we actually just utilize publicly available data in the FluMap. So 45,000 influenza HA sequences. Now, importantly, this model is trained only on sequence data alone, amino acid sequence, and that is all that was given. But because we had all this associated metadata, we could actually, that was available also in the FluMap, we could actually say, okay, let's go ahead and learn the semantic representation, take this kind of high dimensional vector that's coming out of this kind of semantic representation, now project that down into 2 dimensions, and then ask, what does the model learn, and does it have any association with the biology? So this is what I'm showing you here on the left for HA. And so we can actually take this semantic representation, project it down into 2 dimensions by UMAP, and then cluster the data. And then we can overlay this associated metadata. And what you can see is that all these clusters are very much enriched for individual influenza subtypes. Again, just giving us a little bit of confidence that, okay, yes, our language model is learning something that has relevant biology at hand. Now, we also went on to actually take this, take additional metadata and look at host species. And if you actually read the paper, what you'll see is that actually we zoom into particular clusters that are kind of a blend of different species. And in some cases, they're actually associated with some of the kind of avian origins of influenza, for example. So this is really encouraging to us because it suggested that even though we were only passing the model amino acid sequences, that the semantic representation was actually capturing biology that made sense to us. Okay, so this suggests that the semantic representation that the model is learning is associated with at least known biology. Now we wanted to take this one step further, and we wanted to ask, could we actually model viral escape with constrained semantic change search? And again, if I'm a virus, what do I want to do? I want to acquire a mutation that affects the overall structure and fitness of the virus, as opposed to something that's functionally neutral and undergo what we'll call a high semantic change. And 2, I want to be a mutation that does not harm viability or infectivity. So we're going to call that following the grammatical rules. And so that was the hypothesis going in. And so what did we do? So what we did is we actually trained a model on, again, all the historical corpus of HA sequences that were publicly available in the FluMap. And then what we performed is we performed a deep mutational scan in silico of all possible single residue mutations of this specific influenza strain from Perth in 2009. And it's important to note here, again, the model is only trained on sequences, and the model was not actually even trained on any of these mutated sequences. Okay, so this was the model setup, and our hypothesis is that by just training on this model, we'd be able to predict mutations. Now, why did we focus on this Perth strain? And the reason why we did that is there was actually publicly available data from the Bloom lab that actually performed this deep mutational scan experiment in the laboratory setting. And so what they did is they actually made all single amino acid chain mutations to the HA protein in a viral library. And then they took these viruses and then they either performed an infection in the absence of antibody or in the presence of polyclonal antibodies. And then they infected these cells, they read out the viruses by deep sequencing. And what this experiment allows you to identify in this mock selection is here are the viral mutants that retain the ability to infect cells. And then in this antibody experiment, what it allows you to identify are here are the viral mutants that still allow you to infect cells despite the presence of antibody. So this is a really good kind of experimental validation data set for us to evaluate the predictions of our model. So again, our hypothesis is that high semantic change and high grammaticality is going to be associated with viral escape. So we can actually do that experiment and quantify that, and that's what I'm showing you here. So let me just walk you through what you're looking at here. So we can actually perform this deep mutational scan in silico. We can compare to a wild type sequence, and we can say, ask the model to quantify grammaticality of that particular amino acid mutation and semantic change relative to the wild type sequence. And so if we look in this regime that is high grammaticality and high semantic change, first thing let's talk about is this acquisition priority. And really our model is asking us, what we're asking the model to do is acquire mutations on the basis of high grammaticality and semantic change. And what you can see here is that this upper right-hand quadrant is enriched for these red X's. And what are these red X's? These red X's are in fact experimentally confirmed escape mutations. So this was really exciting for us because it suggests that our hypothesis that high grammaticality and high semantic change was going to be associated with viral escape. And so what we were able to do with just sequence alone, we're able to actually predict and validate experimentally determined data. So this is really exciting for us. And the question is did we just get lucky with influenza H1 or does this generalize? And so in fact, it does generalize because we were able to do this for both influenza H3, HIV envelope, and SARS-CoV-2 spike. Now, what was really exciting for us is that even though this model was only trained on sequence alone, what we were able to do further in the analysis was actually say, okay, what if we go back to the protein structure for influenza HA. And one of the things that we saw that was really interesting is that you can look at the influenza HA structure and then superimpose each amino acid with the escape prediction for HA. And what we actually were able to see is that actually there was escape depletion in the stalk domain of the influenza HA protein. And this is consistent with this hypothesis or this data in the field, in the flu field, where actually if you're able to raise broadly neutralizing antibodies against the stalk protein of HA, that's actually broadly neutralizing. And actually work from the University of Washington recently with a nanoparticle vaccine formulation showed that they're actually able to really generate really robust pan flu vaccine kind of concept and idea. So what we're excited about is that potentially these models can not only understand escape, but also make predictions about what may be likely epitopes that would make for really great vaccine candidates. So with that, I think I'll close this second half of the talk. And hopefully what I've been able to share with you is that the semantic modeling approach predicts viral escape in a totally unsupervised, alignment-free fashion, and that the distributional hypothesis from linguistics can be extended to viral evolution. And lastly, that this may enable therapeutic design that anticipates avenues of viral escape. And I guess I'll just give you a little bit of a teaser trailer for what's coming next. So one of the things that we're really excited about is that CSCS, as we call it, has been adopted by the CDC as part of their genomic surveillance program. So this is really, it brings me a lot of joy to see something that was kind of a project that was born out of the pandemic because we were all stuck at home, like have truly translational impact. And the other thing that we're working on is really trying to understand how we can apply these same conceptual approaches to understanding pathogen evolution writ large. Again, really kind of coming back to my first love of TB. So before I take your questions, I just really need to acknowledge the superhero behind the work. So all of the work that I told you today was done by Brian He. He actually just graduated. He's now a Stanford Science Fellow. And he has just been a pioneer of thinking creatively and thinking about what he could do to not have to do experiments. And I have to say he did really well. All of the code is publicly available and we're happy to help. And I also just need to really thank my amazing friend and collaborator, Bonnie Berger, who's a mathematician. And thank you for your attention. And I'm happy to take any questions.
Thank you so much, Professor Bryson. That was an absolutely amazing talk. I absolutely loved all the analogies, and like I've never seen someone present something as complex as like a Gaussian process models, you know, so neatly. So thanks very much. Okay, so we've got what time is it? We've got like 17 minutes for questions. So we'll do our best to accommodate everyone's questions, and I'm going to kind of go through them and either read them out if you've asked them to be read out, or I'll announce your name and then you can unmute yourself to ask the question yourself. And just a reminder, if you still have questions, please send them in the chat to Flavia. Okay, so I think we might kind of kick off with a question from Mark Lipsitch, because he's first on my list. So Mark, if you're happy to ask the question yourself, please go ahead and unmute yourself and ask away.
Thank you. Amazing talk indeed. Thank you for that. I was curious about the sort of battleship analogy and pursuing that a little further. I didn't totally understand, or I didn't at all really understand in the first part of the talk how you do exploration. I understood that exploitation sort of depends on low uncertainty, but you might want to establish a sort of beachhead somewhere else, and then have information in that local area. And I'm sure that's happening, but I didn't get that part from the talk.
Yeah, so we haven't yet formalized how we want to do exploration. What we're doing right now is what we've decided our exploration strategy at present is to say, let's go ahead and take this kind of high uncertainty regime on the small molecule space and really try to find kind of exemplars in each, like cluster that kind of representation of the chemical structures into a set of clusters, and then kind of pick representative members in the kind of unexplored regime to identify, to kind of get some hits out there to try to reduce the uncertainty in that region. So that's kind of our current strategy. But I think there's a lot of ways that we could imagine constructing that. Like all of our kind of acquisition functions at present are literally they're very vanilla. You know, what they are is it's just an upper confidence bound where we like rank in order of affinity and uncertainty, and then we just take 1 plus a weight on the uncertainty. So there's definitely other ways that we could do this, but we've just used kind of a very off-the-shelf kind of vanilla approach.
Thanks.
Awesome, thank you. So the next question is from Leonid Chendelovich, and their question is, to what extent can the features extracted by the language model be interpreted by a domain expert in applications such as the one you presented?
Yeah, that's a really good question, and something we're working on right now. So, you know, like this is in fact the problem with I don't want to say the opportunity with the analogy, right? Is that if I go to my friend down the hall who's a true dyed-in-the-wool virologist, and I say the grammaticality prediction is this, he just looks at me cross. And so what we're doing actually in collaboration with folks is we're actually taking a set of defined mutants where we can actually measure ACE2 affinity and also use a pseudovirus assay to look at neutralization titers, and then really try to understand how we perfectly calibrate affinity and neutralization titer as a function of these two kind of broader linguistic parameters. So that's something that we're working on right now. We don't have a perfect translation at present, but what I can also say is that the Bloom Labs data sets for SARS-CoV-2 spike actually have a little bit more in terms of relative affinities and escape potential because they're more quantitative. And so if we actually just model their data as a proxy for ACE2 affinity and neutralization titer changes, what we find is that our hypothesis that grammaticality is associated with some amount of affinity or infectivity because it's actually well correlated with their ACE2 binding affinity measurements. So we think that that is in the direction, but we definitely are doing more experiments to really definitively kind of explore that more robustly.
And can I ask a follow-up question, which maybe you've already answered and I didn't understand, but so is it possible, complete neural networks and language model novice, but is it possible to pick up kind of any rules from the weightings that get inferred by the model? For example, if there's a role for, say, change in electrostatic charge of like certain substitutions that have an impact, or like location within, like structural location, and also things like which I think you probably you could put into the data, like certain transitions are more likely than others.
Yeah, so those are all really good questions. So what I can say there is that at present we've not we're doing a lot around the weights and amino acid properties. We've not one open question that we've thought about a lot is we were operating with amino acid sequences, but like really mutations are occurring at the nucleotide level. And so how we can think about incorporating that. And then also thinking about, one of the key issues, okay, so the backstory is this project started last year post-lockdown. And the student was like trapped at home and we couldn't think about actual experiments that we wanted to do. And we were really reliant on the generosity of people making data publicly available. And so we were like, we have to be experiment free here. But what we're really trying to do now is really go after all of the biochemistry that we otherwise wanted to be able to do. And I guess the other thing that I'll say is that there is a role if you have additional information to use additional information. For example, like we operated in a structure-free, we operated with no priors, but I think, especially in the SARS-CoV-2 setting, like when we submitted the paper, we actually only had access to 900 sequences with which to train. And so, it's changed a lot since then. And if anybody's interested, we're actually updating the model every 2 weeks with all the publicly available sequences. But yeah, so I definitely think that especially in kind of like information, like sequence poor settings, especially like being able to augment with whatever additional priors that you have would be advantageous.
So it works both ways. We can either learn more with no priors or use our prior knowledge to make predictions better. Interesting, great. So I think this is also a very similar question, but I'll ask it anyway in case you want to approach it from a different angle. So this is from Sam Palace, and she asks, it seems that there are many mutations that are predicted to be highly grammatical with high semantic change, but that are not identified experimentally as escape mutations. Were these mutations not tested experimentally, or is there some additional constraint that prevents these mutations from being functional or successfully escaping antibody pressure?
Yeah, so that's a good question. And that is something that we're definitely reconciling with. Like this was a totally unsupervised exercise. So is there some other dimension beyond semantic change and grammaticality that's important that we need to consider? So I don't think of, for me, I don't think of this as something that operates in an absolute where like, the model predicts that it's high grammaticality, high semantic change, therefore it will escape. But what we really think about this is as a way to kind of prioritize experimentally what pseudoviruses to generate. And so that's what we're doing now, is we're really trying to understand, is there some other third feature to extract that would help improve kind of what we'll call the false positive, so to speak.
Thank you. So this next question is from Xiang Xia. And their question is, in language, certain grammatical components, such as verbs, may have a higher weight in determining the semantics. For viral proteins, it's likely that different regions may also have uneven contributions to antigenic properties. I'm curious about whether a language model could recognize some sort of semantic components from protein regions, for example, regions with high functional importance.
Yeah, so what can I say? Regions with high functional importance. So I don't have that slide. Do I have the slide? I have it somewhere. So I do think that the model can recover important features of structural importance. And you can imagine that it did so for us in the setting of looking at semantic change when we look at the and I found the slide, so maybe I'll just share it really quickly. If I'm
Yeah, go ahead and share again. That's fine.
So, for example, from the structural importance, from the perspective of what is essential, likely for, so this is influenza H1 stock. We can see that there's this escape enrichment that's in the head domain and depleted in the stock. And what you can see here is that for some reason, the stock domain of the protein is not does not tolerate a lot of escape mutation, or like does not tolerate a lot of escape mutation prediction. And a hypothesis there is that that is important for kind of the biological mechanism by which viruses actually kind of like bind to cells. So that's one hypothesis. So I think there's a lot of ways to interpret this observation. One is you could say this is a good region to make an antibody to, or you could alternatively say that this is a region of the protein that cannot be altered in any way that would preclude its ability to be infectious. So I think there's two ways to interpret the data, and I think both are reasonable ways to think about it. So, and I think, lots of people are using language models. I mean, even Facebook has a protein language model at this point, where they're trying to understand important structural features of proteins. So I definitely think that that is 100% a possibility.
So I think related to that, for the example of flu, the stalk is like a kind of, it's just physically inaccessible to, or it's less accessible to antibodies, and certain kind of regions that might be, on a kind of primary sequence level lead to high antibody escape might be kind of irrelevant in a kind of within a host, because once the folding's happened, that kind of epitope is completely hidden. Do you think those sorts of like patterns, I guess it's a related question, those sorts of patterns could get picked up here?
I think yes. I think it really depends. So there's a bunch of different language models that exist. So in the second half of the talk, and I definitely did not make this super clear, like we built viral protein specific language models. In the first half of the talk, what we actually used is a protein language model that was actually trained on like all amino acid sequences. So I think it really depends on how you want to set up the problem. And what you're really looking for. In our case, our hypothesis was that for the second half that, a language model trained on specifically the proteins that we really cared about, would be most appropriate for the problem. But in this first half of the talk, we really just used a language model that was developed in 2019. So I guess my feeling is it really depends on the biological problem you are pursuing. There's no, it feels like the field feels like it's kind of at a stage of its infancy where a lot of things are very much empirical.
Yeah, that makes sense. I think that's the feeling I'm getting from the questions. Everyone's very excited about like, oh, these like biological mechanisms we can pick out. And, as you say, this is such a novel approach. So the next question is from Bill Hannage, who can ask it himself. So whenever you're ready, Bill. You're currently still muted. Okay, I will unmute.
No, I was trying to unmute myself and you weren't letting me. I blame you. So that was very cool. I like that a lot. Thank you. So my question is a little bit an extension of the one that Sam was asking, because I was really struck by the comparisons you were making between flu, HIV, and SARS-CoV-2. And it occurred to me that SARS-CoV-2 right now, all of those things, it's in a very sort of different stage of its evolutionary life. And so the kind of things that you're seeing now following a recent emergence might be somewhat different from what you'd see with a mature association. And so my I guess my question is, as you said, you did this with a very, very small number of sequences. Has the kind of thing you've been seeing been changing at all over time as we reflect the evolving and maturing state of the pandemic? Or have you not looked yet, which would be a perfectly fair thing to say?
We have looked, but I guess our focus has been in quantifying semantic change of the variants of concern, and that's been like what everybody's asked us to do. So that's what we've focused on. We haven't yet focused on like the kind of the evolution of our predictions, or the, it looks like you have a follow-up question as well. So we have not, but we certainly can. So Brian, who's now at Stanford in kind of this like hybrid position, I told him he basically just he took it all. So he's really, he actually has a paper coming out soon, which really tries to use language models to look at evolutionary trajectories. So I definitely think that is like what is in store next. But stay tuned is what I'll say, because I don't want to scoop him.
Awesome. I think let's fit in one more question, because I think it'd be very fitting for our last question to be again from Mark, because he's the director of our center. So Mark, you can have the final question of the entire series.
Thank you. So in the past, people have argued endlessly over beer about whether you should target interventions, vaccines or drugs at conserved areas because they'll have a harder time escaping, or at variable ones because they've shown that they're important targets that are under selection to change. And so it seems like this method actually helps to separate these things out, that you want to target areas where changes would be very ungrammatical and therefore they're rare, but would be very semantically changing, because that's something like you want to force it to do something that it can't do. In other words, you have two dimensions that we've not been able to separate. So that would say like do the stalk, I think. So
yeah, so that's totally consistent with our hypothesis. So, what we've reasoned is that if you're going after vaccine targets, you want to go after high grammaticality, low semantic change regions, perhaps, because those are the ones that are like most likely to be required for some element of like protein folding or binding, or like some function that we can't fully characterize for the virus yet. And then one that is not high, where the semantic change is low. So that's been our hypothesis. And I guess for influenza, there's like orthogonal data to support it. The other thing that's coming out with SARS-CoV-2 is I think people initially were concerned about what regions you could actually generate antibodies against. And so there's some work from the Alter lab and another lab that the name fails me, where like our predictions for SARS-CoV-2 were that the S2 domain of spike would be a target that would be interesting if you could raise antibodies against it. And the work in Galit's lab shows that people with anti-S2 humoral responses have better outcomes for the N that they were able to measure. And that in this other paper, what they showed is that people don't appear to have a problem in generating S2 directed antibodies. So I definitely think that the question of how difficult is it for the immune system to generate a response to some of the predicted regions that we would suggest to target, maybe it's not that difficult. I mean, I'm not an immunogen designer, so I don't want to speak out of turn, but at least I do think that there's natural evidence for these, for people to be able to mount responses to these high grammaticality, low semantic change regions.
Thank you. Well, we're just over the hour, so I think we should wrap up there. But let's thank our speaker, Professor Bryson, again for a fantastic closing talk. And I appreciate people probably need to leave, but I just want to take the opportunity to thank a few people, because this is obviously the final seminar of the series. So firstly, thanks to the Center for Communicable Disease Dynamics and the Blavatnik Institute for supporting the series. Thank you to Benedict Nagnon and Flavia Campanovo for hosting and moderating these sessions along with me. Thank you to Linnea DiPolo for managing the communications, and also to the IT department for helping these talks run smoothly. Thank you to all the amazing speakers of the past few months, of course. And finally, just a massive thank you to Tia Hira and Rachel Sadoff for their phenomenal work behind the scenes to make everything happen. So yeah, thank you again, everyone, for joining. Hope you've enjoyed the series, and have a great summer. Bye.